Hello, everybody. Hopefully you can see me in a minute and hear me. Let's see who joins us then. Right. How are we doing? Have we got anybody there at the moment? Okay, let's see how we're doing. Excellent. Hey, brilliant. I'm starting to hear from people. Can everybody hear me? Just say yes if you can hear me okay, so that I can get on with the class. Hey, Rayan. Yes, brilliant. Yes, you can. Fantastic. I hope everybody's okay and enjoying themselves. Have they had a busy week? What have you been up to this week? Brilliant. Yes. Hi, Wisdom. Moez. Shazia, Rayan, Moez, brilliant. Oh, you like the background. Nice and blue. It's a great colour. So how are we doing? What have you been up to this week? I'd love to know how you're all doing and what you've been up to, and where you're from. So has anybody been doing anything interesting, or has it all been schoolwork? Who's broken up for half term? Has anybody broken up for half term, or is that next week, some of you? From Pakistan, fantastic. Welcome to you in Pakistan. I bet it's a lot warmer out there than it is over here. From London, breaking up next week, break up on Wednesday. That's an interesting day to break up, isn't it? So do you get 10 days, I assume? From Bangladesh, lovely. Zambia, fantastic. I love Zambia. Such a beautiful country. So, and what has everybody been doing? Anybody been doing anything interesting this week? In Pakistan? Brilliant, lovely. Welcome to Pakistan. I started a choir this week, which was interesting because I've just moved house. So trying to get in with the community. So I've just joined a choir. Has anybody else done anything that is new to them this week? So we've got Uganda. Uganda, fantastic. You rode a motorcycle in the desert. Oh my word, that is one of my dream things. It's on my list of things to do, riding a motorcycle in the desert. Always wanted to do that. Really want to go trail riding up the Atlas Mountains. That would be really interesting. Working on a project. What sort of project are you working on? That's always interesting. Right, hopefully we've got everybody. You live in Saudi. Wow, that's somewhere that I have never been. I'd love to go to Saudi. Very interesting. Right, let's get on with the lesson then, shall we? So we've got a lovely web class coming up today, and we're looking at digestive enzymes and metabolism. So let's get this up and running, shall we? So let's share my screen with you. Hopefully this will connect in just a moment and we will be able to start the lesson. Right. Hopefully everybody can see that okay. Right, let's just readjust my screen so I can see it all. Okay. Brilliant. All right, so can you all see that now? Can you see the lead into this GCSE biology web class, digestive enzymes and metabolism? I have to say, I love this subject. So I hope you're going to enjoy it too. I find it just fascinating, really important for us all. Yes, you can see it. Brilliant. So this uses quite a lot of parts of biology, and we're going to be looking at the physiology, and we're going to be looking at the enzymes and what metabolism actually is. So first of all, for those people who haven't seen me before, I will introduce myself. So I'm Philippa, and I'm the lead GCSE biology teacher here at Snap Provise. I have got a BSc in animal science, which is the animal side of agriculture, PGCE in biology and games, and I've just finished as teacher and head of biology, but I have 25 years of experience, and now I'm working with content writing. And hopefully you're going to find that I am enthusiastic as well. So let's have a look at what we're going to be talking about today. So we have these objectives for the lesson. The objectives today are to explain the function of enzymes and the role of the active site, describe the role of carbohydrates, proteases and lipases, our three main classes of enzymes. Ethiopia, fantastic. And describe metabolism and the importance of the building blocks of carbohydrates, proteins and lipids. So how do these build up? What I'm not going to be looking at today is reaction rates. That will be covered in another class. Denaturing of enzymes, obviously, which is a very important part of biology, and tests for substrates, those lovely biological molecule tests. So let's have a look at quickly look at the specification points. So all of you are probably doing different syllabuses, but I just want to quickly look at what we're looking at here. So AQA 4.2.2.1, the human digestive system, and then 2.3 metabolism. So hopefully you're all downloading your specification points from the exam board sites, because it's really, really important that you follow those as you go along in your biology lessons, so you can tick them off when you do them, and then you also can tick them off when you have revised them, so you can keep an eye on where you're at with your notes and your revision. OCR then, we are looking here at explain the mechanism of enzyme action, the importance of sugars in the synthesis and breakdown of carbohydrates, the importance of amino acids in the synthesis and breakdown of proteins, importance of fatty acids and glycerol, and explain the importance of maintaining a constant internal environment. So we're going to be looking at the metabolism and enzymes there. EdXL then, 1.7 and 1.12, explain the mechanism of enzyme action and the importance of enzymes as biological catalysts. So what do we already know? That's a question for us, isn't it? What do we already know? So here we have a diagram of the digestive system. So can we have a look at this digestive system and see what bits we can label? So if I do a little line here, starting at the very beginning of the digestive system, who can tell me what the first part, where we take the food into our body? So we're going to be taking the food into our body. Where do we take that through? Right, brilliant. We've got the liver, which releases, or it's not enzymes. Liver produces bile. That's brilliant. Yeah. So we've got the liver here. The liver here produces bile. And where's the bile stored? It's stored in this thing here. Can anybody remember where the where it is stored? Also, yeah, bile, brilliant. Gallbladder, excellent. So this is the gallbladder. So the liver and gallbladder go together. Okay, so what else have we got here? We've got gallbladder. Gallbladder is found in the liver. So the gallbladder is here in the liver. So that's found in the liver. This here is the pancreas. So we've got the pancreas. Now, the pancreas, this is where some people are getting confused because it's not the liver that produces the enzymes, it's the pancreas produces a lot of the enzymes, doesn't it? Yes, so brilliant. The liver produces bile, which is then stored in the gallbladder, isn't it? Yeah, and they help with the breakdown of lipids. They don't actually break down lipids. They help with it. What they do is they emulsify the lipids to give us a greater surface area for the lipases to work on. Yeah, liver converts to glycogen. We're going to be talking about glycogen. So when there's high glucose, glycogen is stored in the liver and deamination occurs in the liver. We're going to be talking about that today as well. So let's have a think about the digestive system at the moment. Where do we put food into our bodies? We go through the very top bit when you're hungry and you go along and you go, right, I fancy, I don't know, some chips today. Where am I going to put those chips? Yeah, brilliant, into the mouth. So we're going to start with the mouth. And in the mouth, we have a particular gland which helps with the start of the breakdown of starch. Does anybody, yes, we've got the mouth cavity, and there's a gland in here. Produces liquid, yes, salivary gland, brilliant, well done, salivary gland. You are all working really hard here today. What is this tube that carries the food from the mouth down into the stomach? What do we reckon that is? So it carries out peristalsis to help push the, yes, brilliant, the esophagus, very difficult to spell, esophagus, okay, yeah, it can be spelt with an E as well. That's the American way of spelling it. That's absolutely fine. The esophagus, brilliant. And the esophagus carries it down into a very big muscular sac. Yeah, brilliant. The salivary glands release amylase, which helps break down starch. Fantastic. And what's this muscular sac where the food ends up when the esophagus carries it down? Brilliant. The stomach, and you've spelt it right as well. Fantastic. So from the stomach, the food then goes into the first part of this big mass at the bottom. So from the stomach, once it's been mixed up, yeah, we've got the pancreas. Yes, the pancreas also releases insulin, doesn't it, which causes the formation of glycogen. Good. Brilliant. The stomach produces hydrochloric acid and pepsin. Pepsin helps break down the protein. And so then after that, if we just looked at these in general, these are the intestines because they're broken down into the small and large, aren't they? So the food goes from the stomach into the small intestine here and goes past the liver where the bile is released. So the food then goes down through into the small intestine and bile is released from the gallbladder, and that helps emulsify fats, which helps break down of lipids by lipase. So when we go along the small intestine, it goes past the pancreas where all of those enzymes are released that you were talking about. Okay, so it goes past the pancreas, which releases lots of enzymes, also produces your insulin, glucagon. Yeah, neutralizes stomach acid. Good. The bile helps neutralize stomach acid, doesn't it? Because it has alkaline in it. Then it goes through the small intestine. We've got digestion, absorption there, and then it goes into the large intestine where water is reabsorbed. Brilliant, brilliant, brilliant. Yeah, fantastic. And then where's the feces stored before it is digested? Yeah, the duodenum. Good. The ileum and duodenum. Water is reabsorbed in the large intestine. Brilliant. And then it's stored in the rectum. So feces is stored in the rectum before it is digested out of something which is held shut with a sphincter through the anus. Okay, and that's where digestion occurs. Brilliant. Yeah, poop. So, as we've said so far, the liver produces bile, which emulsifies fat. The gallbladder Good, you've got the anus in there, fantastic. The gallbladder stores the bile. And then you've just told me about the bile. Somebody said about neutralizing stomach acid. Yeah, brilliant. So you've also got enzymes being released from cells inside the small intestine as well as from the pancreas. Excellent. And they help with the digestion of lipids, proteins, and carbohydrates. Brilliant. So the bile salts are alkaline, as we said, to neutralize stomach acid. Does anybody know what I mean when I say, and it emulsifies the fat? What does it mean when I say it emulsifies the fats? What does it do to the fat? Small intestine has many villi, good, where you've got the absorption of the food into the bloodstream, makes it easier to digest. Yeah, it breaks sort of large fat globules like this into little tiny ones. So what it's doing actually is increasing the surface area. Yeah, brilliant. So when we absorb those foods, they can be used for things like respiration, etc. We're going to be looking at it. Yes, surface area, large surface area. It gives it a large surface area for the lipase to work on. Excellent. Well done. Really good work. So bile is it's like a green solution that the liver makes. And so what it does is it uses substances in the liver to produce this sort of green substance, which has this alkaline side of it, and other bile salts, which emulsify the fats. And some of those come from broken down red blood cells. Bilirubin and biliverdin give it its color, which are they're colored substances from the breakdown of red blood cells as well. So there we go. Moving on then, we'll look at part 1, enzymes, because obviously we need to know all about the enzymes, don't we, when we're talking about digestion. Let's have a quick look of the time here. Right, so enzymes. What are enzymes? If I ask you to describe to me what an enzyme is, what are we going to go for? It's the bile which is produced by the liver, which is then secreted. It's that green solution which is secreted into the small intestine, and that's the bit that emulsifies the fat. So the liver makes the bile and the bile digests the fat. Okay, good. So it is a catalyst. Bile is not an enzyme. Okay, that's where people do get confused. Bile is not an enzyme. It's just a solution which helps the enzymes work by giving a larger surface area. So it's bile that emulsifies the fat, not the liver. Right, brilliant. So enzymes are biological catalysts, as you've already said. Biological catalysts. Are they used up when they carry out the reaction? Are enzymes used up or changed in a reaction? Does anybody know? Yes, they speed up the rate of reactions. Okay, brilliant. Proteins, or you could say they're polypeptides. Okay, and made of polypeptides, and their overall shape gives them their proteins. No, they are not. They're not used up. They're not changed. So they're not used up and not changed in shape or form in the reaction. In other words, they're reusable. Excellent. And they speed up the reaction without getting used up. Good. So they're biological catalysts that speed up the rate of reaction without being used up in the reaction. All right. And they do this by lowering the activation energy. In other words, the amount of energy that needs to go into the reaction for the reaction to occur. Yes, good. They will get, the reaction doesn't change them or denature them. It's changes in other conditions that could denature them. Well done. Yes, they provide an alternate pathway. Excellent. That's how they lower the activation energy. Good work. Right. Let's have a look then at how enzymes work. So first of all, what happens if well, if the substrate doesn't fit in, either it's the wrong substrate or the enzyme has been denatured. So each substrate has its own particular enzyme, which is going to catalyze the reaction. All right. So all the enzymes, and that's what we're getting onto now, all enzymes are specific to their particular substrate. So they can only catalyze one type of reaction. All right. So that's it. Yeah, you've got to have the perfect match of the substrate. So how is the shape of this active site formed though? So we have an active site, don't we? All right, so here is an active site, isn't it? So this is an active site. How is the shape of that active site formed? How is a particular enzyme specific to a particular substrate? Well, that's what we're talking. Well, we're talking about the lock and key theorem. So we're looking at, this is a proposed explanation for scientific observations to explain the action of enzymes. All right. So we try to use this. This is a basic theory, and at A level, you go into a little bit more of the theory. All right. So this is the basic theory, the lock and key theory. All right. So can anybody tell me how the shape of an active site is formed, thinking about the structure of proteins? What are proteins made up of? What is the independent, each single unit in? What is the single unit of a protein? So the building block of proteins. Can anybody tell me that? Begins with A, AA. Amino acids. Excellent. So each protein is made of a string of amino acids, isn't it? Okay. So the string of amino acids can be, I mean, you've got 20 different amino acids, so you can have a huge variety of chains of amino acids. Good. So, but the actual shape of that active site is formed because of the unique pattern of amino acids along the chain. So each amino acid chain, all of the amino acids interact with each other, they form bonds with each other, they interact with the surrounding environment. So the overall shape is the shape of the enzyme. It's a 3D shape or tertiary structure, all right? And the active site is formed because of the unique pattern of amino acids along the chain to make the protein, because they all interact with each other and form bonds. Okay. The substrate is what goes into the active site. Yeah, the formation of an enzyme is super complex, and it's all coded for by your genes. All right. So your genes determine which amino acid goes where, and then which amino acid goes where determines the overall shape of the protein. All right. So what is the active site then? Having talked about that, what is an active site? So yeah, the formation of enzymes is super, super complex. So what is an active site? What fits into the active site? Somebody told me earlier. It's the part of the enzyme. I will talk to you about what, yeah, it's the unique shape. So it's the part of an enzyme. Good. Where the substrate binds. Excellent. Where the substrate fits into. And what it does is it either brings two substrates together, all right, or it can break them apart. So it can work on one substrate to break it apart, or it can work on more than one substrate to make bonds in the substrate so that it fits them together. So you could either be building up a new, chain of DNA using DNA ligase, or it could be breaking them apart like amylase breaking up starch. Okay. So the idea is it's a lock and key theory where the substrate is the key that fits into the lock. So the active site is like the lock, and then you've got to find the right substrate to fit into that active site. So for example, this one here won't fit, But this one here is going to be the right substrate to fit into the lock. Yeah, amylase and catalase. Okay, so catalase breaks down hydrogen peroxide into water and oxygen. Good. Yeah, exactly, with the error in the DNA. Good. So these are the keys here which fit into the lock. So it's this one that's going to fit into the lock. And when the enzyme and substrate come together, they form an enzyme substrate complex. Let me just see what you've just asked. Well, if one of the amino acids is changed, then that can lead to denaturation of the enzyme. Okay. So enzyme substrate complex, then the products are formed. So you could call that the enzyme product complex. And then the products here are released. So they've changed slightly in shape. They're not the same shape as the original substrate. The substrate is what goes into a reaction, and products are the things that come out. Okay. So this could be an example of, say, sucrase breaking down sucrose into glucose plus fructose. So that could be sucrase breaking those down. So we have enzymes and substrate sort of around in solution. They bump into each other. And if they bump into each other in the right angle, then the substrate can fit into the active site and the enzyme substrate complex is formed. Then what happens are the products are produced and they're released. All right. Products are released and then the enzyme can be reused again and again and again and again and again. Okay. They may produce less enzymes, yes. As you change age, the quantity of enzymes that you produce can either increase or reduce. All right. So there can be and some people can stop producing certain enzymes such as lactase, and that's why people become lactose intolerant, because they can't break down the lactase. Enzymes are not destroyed by the reaction. They can just be reused again and again. They can be destroyed or denatured if the surrounding environment changes, such as the temperature increases or the pH changes. All right. Right, we better get on because there's loads of questions. I'm loving this. Okay, let's have a look then. Digestive enzymes, some examples of digestive enzymes. What have we got here? Can anybody tell me what the name of enzymes A, B, and C are? I find it useful when I put letters up and then you can just put a letter for the one that you know. Can anybody give me the overall names for these type of enzymes? Amylase is an example up here. So e.g. amylase. It's breaking down carbohydrate. Good. Protease. Lipase. And this one, it's always A's, so it's carbohydrates. Good. Yes, lipase breaks down lipids into glycerol and fatty acids. We've got that here in the last box, haven't we? Good. So whereabouts are they found? I'm going to put letters in again. Where are they produced? Good. Protein into amino acids. Carbohydrates break carbohydrates down into more simple sugars such as disaccharides and monosaccharides. Lipase in the small intestine. Good. Pancreas, small intestine. Good. Salivary glands. And pancreas into small intestine. Good. What about protease, such as pepsin? proteases in the stomach, and also somebody told me earlier, and the small intestine. Good. Pancreas into the small intestine, and the small intestine also produces many of these as well. So food is broken down by the enzymes into small soluble molecules that can be absorbed into the bloodstream. Somebody was talking about bile earlier. Well done. So generally, as you can see, enzymes end in the 3 letters A, S, E, A, S, E, A, S, E. So if you see A, S, E, it is normally, it would be an enzyme that you're looking at. All right. So let's now have one more look at the pancreas before we think about an exam question. So pancreas, enzymes are released from the pancreas into, and you've all told me actually, into the top of the small intestine where digestion occurs, because it's the second part of the small intestine where absorption occurs. All right. So from the stomach, so it goes from the stomach into the small intestine. It goes here, past where the gallbladder is here, which releases the bile, which is not an enzyme. All right. And this is the pancreas here. And so it releases it into the duodenum, because it's the ileum, duodenum, it's the ileum where absorption occurs. Brilliant. Right, how are we all feeling about this? 1, 2, or 3? Straightforward, all right, super confused. How are we feeling about this? Are we ready to do one exam question? No, stomach is bigger than the pancreas. Yeah, you're all happy with this? Good. Are you a bit confused? Right, do ask questions as we go along if you are super confused, all right? Okay. Right, this is an example of an idea of the lock and key mechanism, isn't it? This is showing the lock and key mechanism again here. The stomach is larger than the pancreas, all right? The pancreas is smaller than the stomach. Stomach fits quite a bit of food in, all right? So let's have a look at this. Use a diagram to help to describe the theory of enzyme action. So can anybody tell me a bit that we can put in? Can you please turn off slow? I don't know what the slow is. How long does it take for digestion to be completed? That depends on what you've eaten. All right. So food normally stays in the stomach for a few hours. All right. So let's have a look at this. Enzymes. How are we going to describe the enzyme action here? It's 4 marks. What are we going to say here? Can anybody tell me anything that could go into this? Anybody tell me? Can't start. Can we start off by saying this is the lock and key theory and label the substrate and the enzyme? Substrate and enzyme labeling is a great idea. So substrate, enzyme, good. So what we're saying then is, and where does the substrate fit? They fit in perfectly. They're complementary in shape. That's a good word to use, complementary in shape. Fit. Where does it fit in the enzyme? What do enzymes have? Good. The active site. Good. Enzymes have active site. I thought you were confused there, Aaron, but that's really good. Well done. So enzymes have an active site. That would give you 1 mark, which is complementary to a specific substrate, isn't it? So we've used that word complementary. What is formed here? So that's stage 1. What's happening at stage 2? You can make notes, or you can always go over this again, because this is going to be available on YouTube now, so you can go back through it if you want to, which is better. So talk to me now, and then make notes later if you want to. Good. It's the enzyme substrate complex. The enzyme substrate complex is formed. And that gives us 1 mark. And it fits together, as you said right at the beginning, fits together like a key in a lock. So there's another mark. Okay, so that's super. So, and then you could say, if you didn't fit that last bit in, you could say the products are released and the enzyme is reused. That's another 1 mark. Enzyme substrate complex. A complex is a formation. So it's basically saying that the substrate and the enzyme have joined together to make a new formation. That's what an enzyme substrate complex basically means. It's not, they're not that they are complex, they're forming a complex. So, you know, when you talk about a building complex, it's the shape of all those buildings together. All right. Yeah, exactly. Please do ask as we go along, because that's what I'm here for. That's the whole point of this session, is for me to explain all these queries that you may have and maybe don't get time to ask at school. All right. So there we go. Well done, everybody. And I'm really glad that we cleared up a few things. Yeah, complex of the formation. Right, let's now see how all this fits in with metabolism, shall we? Forming a complex means it's forming a new shape. All right, so a complex is the shape of things together. So an enzyme coming along and joining onto a complex, onto a substrate, that forms a new shape, doesn't it? And they're working together to form a new shape. Okay, so let's have just better move on to metabolism, because we're running out of time because you're all talking so much, which is absolutely fantastic. I'm really pleased that you are asking so many questions. So with your one that you asked, the active site binds to the substrate, good. So the active site is the part that the substrate binds to. Each substrate binds to a specific enzyme in order to convert substrate into smaller chemicals. So as long as you said, if you put in that they are produced into products and products are released, that would be a mark. So 1, 2, 3, yeah. If you would put in products, you would have got 4 marks on that one. All right. So let's have a look at metabolism then. Metabolism, what is it? Can anybody tell me what metabolism is? All the chemical processes, brilliant, in the body. So it's the sum of all the reactions in a cell or body. Good, brilliant, well done. What about examples? Give me an example. A classic one that releases energy. It's controlled by enzymes, good. Which metabolic reaction occurs to release energy? Good, respiration. So an example is cellular respiration, which releases energy. Happens in the mitochondria. Good. All right, so energy released from respiration. And then what happens is that energy, that ATP, then the energy is used for other metabolic processes. So it powers other metabolic processes by providing the energy for them to occur, to break down and synthesize molecules. So if it wasn't for respiration occurring, there wouldn't be enough energy to power all these other metabolic reactions. Good, so excretion is needed. So converting excess proteins, deaminating them, brilliant, okay, and it occurs in the mitochondria. So enzymes are required for all of these metabolic reactions, and metabolism is a function of homeostasis, which is how we maintain a constant internal environment. So if we didn't have metabolism and enzymes, we wouldn't be able to carry out homeostasis. We wouldn't be able to control our internal environment. So with this one, if you see, what we've got is we've got the substrate going in. Okay. The product is formed. So we've got a product formed here, which then goes into another enzyme. And this time, this one, it was breaking it apart. This product, which is formed from breaking this first substrate apart, then goes and joins with another substrate here. And that binds them together to make another product, which then goes into another enzyme. So what you have is each product that is produced from one reaction goes into the next reaction, which goes into the next reaction. And it's this massive chain of reactions that's going on. And that's your metabolism. Yeah, balance in the body. Yeah, because starvation, you haven't got, well, it's not just for respiration. With starvation, it could be that you also haven't got enough products to produce proteins and things like that. And you can have heart failure and all sorts of things because you're missing those molecules which are needed to make new substances in the body. All right, so starvation is a little bit more complicated. But one substrate will bind to one enzyme. That enzyme then produces products, which then go in as substrates to another different enzyme because they're a different shape. All right. So you get the products, which are a different shape to the original substrate. So they now become substrates to the next reaction, which then produces more products. And those products then become substrates in the next reaction. And so it's this sort of chain of reactions when the product becomes the substrate in another enzyme, which produces products which become substrates in another enzyme reaction. And so it carries on. Okay, it's a massive great chain. So they are specific. Enzymes are specific to only work with certain substrates. So molecules can be broken down, such as salivary amylase breaking down starch, or so in the first one, say that could be maltose joining maltase, which produces glucose molecules, which then go into another reaction. All right. Does that kind of make sense? It's a very complicated topic. Let's have a look at the next thing and see whether you're happy with the next part. So do ask if that, does that make sense? Okay. Do ask, carry on asking. So let's first look at carbohydrates and the uses of carbohydrates. What is glucose mainly used for? What is glucose mainly used for? We've just talked about it. We just mentioned it, actually. Yeah, to produce ATP. So it's used in cellular respiration. And it produces ATP, doesn't it? So that will produce ATP. It can also be used to make amino acids. ATP is the molecule which holds energy at the end of respiration. It's like a little it's a substance which has very, very high energy bond in it. And when that high energy bond is broken, it releases energy which can be used for muscle contraction or whatever. Okay, so ATP is basically a packet of, it's a tiny, it's an adenosine triphosphate. Triphosphate means 3 phosphates. And in that last phosphate that's held on, there's lots of energy. Okay. So glucose is made in photosynthesis. We have to remember that photosynthesis is really important for us. If there weren't plants, we'd have no food chains. All right. So energy is stored in ATP. Hopefully that makes sense. So do ask if that doesn't make sense. So glycogen, we've already mentioned, glycogen is stored in the liver, isn't it? Storage in animals, mainly stored in the liver. So glycogen is made by the liver by building up glucose molecules and joining glucose molecules together. It's also stored in muscle cells. ATP stands for adenosine triphosphate, which means 3 phosphates. ATP, you need to know that ATP stores energy, but you don't need to know its full name normally. All right, you don't need to know its structure. That is A level normally, okay? So, but glucose, glycogen, starch, and cellulose are all very important, so can come up in your exams, all right? So starch, this is a storage molecule again. Where is it stored? In what organism do we find starch? Think about all the carbs that you might eat. Plants, excellent. Storage in plants, thinking about potatoes. It's insoluble, just like glycogen, so it's not going to affect the osmotic potential. In other words, it's not going to be soluble in the cell and draw water into the cell, because that would be disastrous, because that can cause lysis of the cells. Okay, good. So cellulose, where do we find cellulose? Again, it's in plants. What part of plants does cellulose make up? Cell wall, excellent. So cell walls, we can't digest cellulose. So it's also important with the growth of the plants, obviously, because they need to lay down more cell wall. All right. So it's found in cell walls. We can't digest it, but herbivores have bacteria in their gut that can digest it. Any questions about those? Was that all relatively straightforward? Is that familiar to you about the carbohydrates, hopefully? So we'll move on to proteins then. Let's have a look at proteins, because we've just got proteins and then lipids to look at. All right. How many different types of amino acids do we have? It's good bacteria that break up the cellulose. Good. Yeah. There are 20 naturally occurring. Okay, so amino acid gets its name from the amino group and the carboxylic acid group of their structure. I think some of you are thinking about DNA and the types of nitrogenous bases there. So there's 20 naturally occurring amino acids, which gives us that huge variety of amino acid chains, which give all those different shapes to the structure of the protein. All right. Some of these amino acids are essential amino acids. Some of you might have heard of that, especially if you are a vegetarian or vegan. You might have heard about the essential amino acids that we have to consume in our diet. And that's why they are there in meat, and it's harder. We've got to balance it as vegetarians or vegans to make sure we get all of those in. 20 amino acids, it does come up. You probably may need it for GCSE. It's good to mention it, but the idea that I'm telling you about 20 amino acids is the fact it gives you the idea of if you have 20 amino acids and you have hundreds of amino acids in a chain, it gives you huge different structures in proteins. All right. So some of you don't need to know, but they are made from glucose and nitrate ions, and you know this more from plants, and the fact that plants photosynthesize and produce glucose, then they need to get the nitrates from the soil to add to the glucose to make the amino acids. Okay, phospholipids we're just about to come on to. All right, so this tends to come up more when we're talking about plants and photosynthesis, and then how that glucose is used from photosynthesis, and the fact that glucose and nitrates give us amino acids. So that's why the plants can then grow, because they've made their own glucose and then they've absorbed nitrates from the soil. Okay. In the bit of the human physiology, they are broken down into urea, and that's by the liver. All right. Yeah, plants have amino acids. They need to grow. They still need proteins and lipids. All right. So they produce carbohydrates by photosynthesis, but they then have to convert those into lipids and proteins. So they have to get the nitrates from the soil to make those proteins. So this diagram here is trying to explain what I was talking about before, about these long chains of amino acids, all right, of 20 different types of amino acids. So when lots of amino acids join together, they make a large polypeptide, which is a polymer. Poly meaning many, peptide is an amino acid joined to another amino acid. All right, so short chains of amino acids are peptides. So polypeptides means lots of amino acids joined together. Yeah, so amino acids are the basis of all proteins, right? So all proteins are made of amino acids, and all living organisms need proteins, so they need the amino acids. Okay, and they're joined together by peptide bonds. Brilliant. Now, when these polymers, do you remember me saying that amino acids form bonds with each other? So as soon as they come near each other, they can form bonds, and that gives a sort of 3D shape to the protein. So they start to fold up, forming these bonds, and that's when they're called a protein. So a protein is one or more polypeptide chains forming this 3D structure. All right. So this is a simple protein, which is just one amino acid. Amino acids is a monomer. Brilliant. Yeah, you do need to know that amino acids make polypeptides, which make proteins for just about all courses. All right. And then one protein can join on with another one, or more than one. Okay. So for example, this is hemoglobin here. And this is 4 chains of polypeptides, so 4 simple proteins joined together to make a more complex protein. So this is a protein made of multiple polypeptides. So this is a more complex protein. So this is hemoglobin. Okay. Well, this is a primary structure here. Yes, that's a primary structure. So that's the first structure. It's just the long chain, isn't it? Most of the time, primary, secondary, tertiary is really just needed for A level. So we're not going to talk about it. The secondary isn't here. No. The simple protein is your tertiary structure. Tertiary structure, quaternary structure is the end one. Okay. But most of you don't worry about that. All right. So let's move on to the uses of proteins then. So we've already talked about one. So monomer is monomers joined together in the chain. That's the primary structure before it starts to fold. So don't get those confused. That's why I don't really want to talk about primary, secondary, and tertiary structures. All you need to know is that monomers is a single unit. That's a single building block of all biological molecules. So an amino acid is a monomer of proteins. Polymers are when lots of monomers join together. That's all you need to know. All right. So one monomer, like amino acid, joins to another one, and eventually you've got a polymer. All right, which is lots of single units joined together. Mono meaning one, mer means unit, so one unit. Polymer, poly means lots, mer units, so that's lots of units joined together. All right, just stick with that for now. All right, cool. So this first structure here, what is that? We've just been talking about it earlier on. What's this structure? Begins with E. We've just been talking about it. Yeah, brilliant. Enzymes. Now, this is hemoglobin. Can anybody tell me what the role of hemoglobin is? Good. Yeah, the enzymes forming the locking key. Brilliant. This second one is hemoglobin, carries oxygen. Brilliant. So some proteins are carrier molecules because they carry things around the body like hemoglobin. Then we've got things like collagen, all right, in our skin. What can anybody tell me? What does anybody got any idea what collagen is? You find it in all sorts of parts of the body. So these are known as structural proteins. Yeah, hemoglobin is found in red blood cells and it carries the oxygen around the body. This is a structural protein. They tend to be long, thin fibers which make up things like your tendons, your ligaments and things like that. Yeah, blood vessels, walls of blood vessels are made of structural proteins. Bones contain structural proteins. Excellent. Good. Well done. And then something that helps regulate the whole body, things like adrenaline, insulin, glucagon, ADH. What are all of those? Begins with H. Estrogen, brilliant, hormones. So many hormones are also proteins. I'm just keeping an eye on the time because we've got one more to do, which is lipids. Does anybody recognize this structure here? So lipids are fats and oils and they're insoluble in water. Right, brilliant. This is a triglyceride. Okay, so this is an example of a triglyceride. Glyceride comes from the word glycerol, so it's a glycerol structure, and it has good, well done, 3 fatty acid tails. Okay, and the main part of that is C, 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 and they've just got lots of H's off them like this. Those are all H's, and then it has a carboxylic acid group at the end, and those are your carbohydrates. So those are your lipids. So, and there's your carboxylic acid, so that gives you the acid bit. Fatty acids. Where are they found? Somebody was talking about phospholipids. Glycerol is 3 carbons with hydrogens on. Okay. And OHs. So glycerol is just a simple structure made up of, it's a 3 carbon structure, which then binds to 3 fatty acids. All right, and that's part of lipids. Cell membrane, excellent. And they're also used as a chemical store of energy. As fat in the adipose cells. Phospholipid bilayer, excellent. Yeah, good, well done. C3H8O3. So lipids, the main role is either in cell membranes or for energy store. So how does everybody feel about that? Are you all happy with that? Okay, we have 2 minutes, so I'm just going to ask you, see whether we can quickly do this. All right, so metabolism is sum of all the chemical reactions in the cells of the body. One metabolic reaction is the formation of lipids. Give one other metabolic reaction in cells. It was one that we were talking about, is a classic. To release energy. Yeah, for heat regulation, that's good. So heat regulation from shivering, which requires this respiration. Excellent. Respiration to release energy. Good. All right. And then we've just quickly got this. The table gives information about food molecules. Complete the table. So what are carbohydrates? What's the monomers? We're looking for the monomers here. What are the monomers of each of these? Good, glycolysis is another one. Yeah, sugars, monomers, it's the, you can put sugar or glucose, that's fine. Here was the fatty acids, good, and glycerol. Here were the good, the amino acids, those are the monomers there. And here the main function is respiration. In other words, an energy source. Well done, everybody. That was very quick, that last bit. So hopefully now you know about enzymes, the active site, carbohydrates, proteases and lipases, and metabolism and the importance of the building blocks of carbohydrates, proteins and lipids. That was a lot to cover in one lesson, but I think you've all done really, really well. Do make sure you set a reminder because of the next YouTube coming up. Everybody go and have a good rest of your day. And I hope that I'll see you again next month. All right, it'll be really nice to see you. So thank you all for coming. Thank you for participating so well. It was fantastic. All right then, bye-bye. Thank you.